Hey everyone, how's it going? I'm in Italy. Can you see? Hopefully you're doing well. You had a nice weekend. Um, I think we can get started right away and talk about your exam that you have coming up this weekend. So let's go ahead and do that. Just a second. Okay. Um, Let me share my screen. Recording. Okay, hold on just a second here. Get myself set up. Good. Good. Okay, so you have an exam this coming weekend. It can be taken any time between Friday, uh, 12 a.m. if you like. So the exam will be up at midnight Thursday night, and it will be available through Sunday at midnight. So any time during that 3-day period. It's a 90-minute exam. I think there's going to be about 60 questions, give or take a few. I haven't refined it yet. I've got to talk to the discussion section leader, your TAs, to find out which questions we can post. But it'll be about 60 questions, multiple choice. And it is timed. So once you start it, you have 90 minutes to finish it. You can't stop in the middle or pause or anything like that. If you have to run to the restroom, your clock's ticking. But you should have more than enough time. I mean, it should be no problem. We've certainly been able to do this in live settings with the live class, and we've done this also with the remote class. So it should be no problem. No one's ever had a problem finishing it. So that's the details of the time. As you know, it is open book, which is nice. You'll have full access to your slides, any notes you've taken, the lecture videos if you wanted them, your book. However, and again, this should be pretty obvious that 90 minutes is not 3 days, so you cannot look up every answer. There's just no way you're going to have time. You absolutely need to come in to the exam having studied. And that's very important. I know that whenever people see open book exam, they just assume that they don't need to study, but that is not true in this case. The way you should take this exam is you should study hard. You should study before Wednesday when you'll have a chance to ask your TA some questions in discussion sections. So you at least should do some of your studying before then. And then more studying between Wednesday and the weekend. And you should really be pretty much prepared for the exam as if you were taking it in class and it was closed book. The advantage, though, is that after you go through the exam and answer all the questions you can, having studied so well, is that you can go back and check your answers along with the notes. So you should think of this as an open book exam in the sense that you can clarify any questions you've had problems with. Again, I want to emphasize this. You have to come in having studied or you're not going to do well. So people generally do very well because they do take that advice. But every once in a while, we have students who haven't studied and they do disproportionately worse just because they didn't have time to look up all the answers. I just want to make sure that that folks don't do that this time. So please do study. As I said, people do well, so we don't have to really curve very much. Typically, if you get so 80 to 90% would be in the B range, where we would, also assign B minuses and B pluses in the long run. But you'll see how we do this. We don't assign any firm letter grades until the very end after you take the final exam. But think about it, you get 80 to 90%. That's the general B range in terms of percentage correct. Okay, we don't add a curve when we have an open book test like this. Generally, we don't need to. Okay, even though it's open book, it's very important that that you take the exam on your own. You are not to take this with any friends over Zoom or something like that. right? We can usually determine that if when it happens, and it hasn't happened in a long time, but there are ways for us to figure that out in terms of the patterns of incorrect answers and that sort of thing. So this is a test you should be taking on your own anytime in that 3-day period. But again, it is on your own, despite it being open book. It covers everything through taste perception, which I believe I might be able to finish today. If not, I'll certainly be able to finish it Thursday. Oh, I put most. Well, we'll see. But it will be very clear when I'm done with the taste perception material, and on Thursday I'll certainly start with touch, but that material will be on the final exam, not the midterm. All your discussion section material through Wednesday, and actually that's going to be a little on touch. You'll see. So there will be a little touch material on the final the midterm exam. I'll talk more about that on Thursday. We'll see how much of the material the section leaders, the TAs, can get through on Wednesday. But all book material through chapter 5, including the preface. And so there are at least 2 questions on the exam, as I recall, that are derived right from the preface. And I did not have a chance to discuss that material in lecture, and we didn't discuss it in the sections either. So make sure you go into that preface and you understand some of the concepts. There's not a whole lot of material that's test worthy in there, but it's important that you go through and you understand what the preface is discussing. Okay, and then finally, as I just mentioned, you will have some time on Wednesday to have your questions answered for the exam. So the section leaders, I think, are going to leave about 10 minutes to ask questions on Wednesday. And again, what's important is that you come in with questions. They're not going to do reviews. They're not going to, kind of go through the slides and go through the lectures one by one and highlight the important points. Okay, they are there to answer your questions. Therefore, it's important you come in with questions. So you should do some of your studying before Wednesday's sections. Okay, if you don't come in with questions, they'll say, good luck, see you next week, and you won't have a chance to have your questions answered. I'll put up a couple of practice questions. It's going to be probably 5 practice questions, and they are not a study guide. It's not a way for you to kind of organize your studying. It's just a way for you to understand how the questions are asked. They are multiple choice, but because there's a lot of material and we need to test your understanding, they're going to be answers in the multiple choice exams where answer D is all of the above, or answer D is none of the above, or answer D is both A and B are correct, that sort of thing. So it's important you read all the answers thoroughly, even if you think, oh, it's A because that's the first answer you recognize, doesn't mean that B is not also true, in which case D might be the correct answer where it says both A and B are true. You've had these type of questions before, but I just want to post a few so you get a general sense of the format that we're using. Okay, any questions about the midterm exam? There is a lot of content in the book. That's very true. I would concentrate in the book on the material that I did not cover. Okay, so there's overlap, certainly, but there's going to be material from the book that I did not have a chance to cover in section or in lecture or in section, and I would concentrate on that material. You're not going to be, a lot of the book is, discussing the folks I interviewed and, the details of their background. I'm not going to test you on those details of the interviewees or the interview, process, where it was, or anything like that, right? The interviews are really there for you to be introduced in a fun and kind of engaging way in the concepts. So none of the details about the interviews are going to be tested. I should say this, that, I might say, an echolocator such as Daniel Kish might be able to do this, but I'm not asking about, Daniel, a specific about Daniel per se. It's just Daniel as an example of an echolocator. Okay, so it is, much more on the content of the ideas. It's much more conceptual in the sense that I won't be asking details of any of the people I interviewed. Okay. Other questions about the exam? Okay. I should say this, that, the way I generally make up the test is I derive most of the material from the lecture. Of course, there's a lot of overlap between the lecture and the book, which is good for you. And, it's a non-majors course, so it's we try to make it straightforward. But imagine a good percentage of the questions are going to be from the lectures, and then a smaller percentage will be from discussion section content and book content that was not covered by the lectures. Okay. Can you list the chapter that you didn't cover? I think, I'm not exactly sure what you're asking there. I think we're going to be covering everything through chapter 5. That's taste. Let me see. Let me look to see if there's any material on touch at all on this first exam. But I'm certainly, you'll be responsible from the preface through chapter 5. I don't know if that answers your question. Is there another way that you're asking that question that I can help? So it's everything from the preface to through chapter 5. It is, again, am I not answering your question? It's there in the slide there. Hopefully you're seeing that. Chapter 5, all book material through chapter 5. Maybe I'm not understanding your question still. I apologize if I'm not. Okay, I assume now that you understand that. Now you understand. I'm answering it. If you come up with another way of asking that because I'm not understanding it, please do let me know. Okay, other questions? Other questions? Okay. So what we're going to do today is finish our discussion of smell and then get into taste. And just to give you a kind of a reminder of smells. I think smell is like so cool. And we went through this last slide. This is the last slide we went through. And as we were discussing smell, we started discussing some of the kind of conscious sort of smell experiences we have, and some of the experiments that have tested our kind of conscious smell, in the sense of being asked specific things about how something smells, and then how we might recognize things like our relatives and friends and things like that. But then we spent a lot of time, the second chapter on smell, chapter 4, spends a lot of time talking about unconscious influences of smell. And to remind you of that, much of our smell processing happens when we're not conscious of smelling something, right? So when we first walk into a room, we might smell, something that's cooking, or some perfume that somebody's wearing or something like that. But we relatively quickly lose our conscious awareness of that. We kind of adapt to that smell, and we no longer have, kind of a conscious recognition of that smell. But our brain is always processing smells, and all the smells in a room, and some that are never even conscious to us, without our awareness. And that's what we were talking about. And what that led up to was this idea that there might be kind of some type of chemical communication that goes on between people through our smell system. So we talked about the very famous research of Martha McClintock and her assertion that menstrual entrainment happens with humans like it does with many other mammals, and that entrainment, like it does with mammals, occurs through our scent system. Right now, again, this is a controversial issue still, We're working on that controversy a little bit, but she believes she has pretty solid evidence that by applying uh, natural body odor secretions to other people, she can show that menstrual cycle timing can be changed depending on if a receiver is getting the smell of an ovulating woman or a pre-ovulating woman. That seems to pretty consistently shift the menstrual cycle in one direction or another. She believes that's good evidence that, yeah, there is something to be said about human pheromones. Now, what the part of the concern is about that is that the type of pheromone influences we see with animals are more than this just the menstrual entrainment, more than just the kind of matching up the cycling of say mice that are cohabitating the same nest, right? Some of these sorts of influences seem to induce a reflexive, a very kind of abrupt, conspicuous, reflexive response in an animal. And we already talked about some of these, right? We discussed the ants that are smelling a fearful scent from another ant that causes them to basically reflexively run away from the location where that smell was emitted, right? And so it seems that when they are exposed, when an ant is exposed to that smell, it is something they can't help. They need to run away. That doesn't really seem to be the case when humans are exposed to a fearful smell of another human. Now, we did talk about some pretty cool effects where, it seems that humans can be kind of put in a state where they're a tiny bit more jumpy, a tiny bit more fearful, but it's very subtle, right? You're exposed to a person's smell who's been scared, whether they're scared through a roller coaster ride or right before they're taking an exam. That exposure, that unconscious exposure to that smell can make our eye blink response, our startle response to a loud noise, a little bit more speeded and a little bit kind of greater, a little bit more amplified. However, without that loud startle sound, okay, our eyes aren't going to blink because we've just been exposed to the smell. It's the sound that induces the startle response, the eye blink startle response. It's just that when we hear a loud sound and are also getting this unconsciously presented smell of somebody who's scared, the startle response, the eye blink response is a little faster and a little bit greater, a little bit more amplified. Okay. What else? Well, it seems to make us a little bit more vigilant on exams. It can seem to, have some influence over what else did we talk about? Let me just make sure. Oh, here. Yeah, facial expressions. That's the other one I talk about in this context. Yeah. It makes us interpret neutral facial expressions a little bit as a little bit more negative. Oops, sorry. But when we're exposed to these fearful smells, we don't run away, right? We barely have any notice of it at all, probably. It just kind of shows up in very subtle ways in these other sorts of tests. So we are not reflexively influenced by other animals' pheromones. Doesn't seem we are. Okay. And then the other one we talked about was the pig stance. If a female pig seems to detect, a female pig in estrus seems to detect the odor of a male pig, they seem to kind of reflexively go into the mating stance. That's nothing like what happens with humans at all. Okay. What Martha McClintock and other researchers have suggested is that we need to kind of rethink what pheromones are like for humans. So she'll say, yes, the pheromone influences for humans can be more subtle, certainly, but they are still pheromone influences. And so that's where we left off. We talked about different types of pheromone influences that are discussed in the animal world. One is the primer influence, and that's the ones that we've discussed that induce a menstrual entrainment, which also might happen to some degree with a man scent. So there is some evidence that if a woman moves in to cohabitate with a man, and I guess it's true whether or not she's romantically involved or not, the presence of a man will kind of regularize, meaning it will make her menstrual cycle more consistent, and it's thought that might be done based on smells, unconscious smells from the man. It's not completely clear, but that might be another example of a primer pheromone. Okay? But releaser pheromones, the ones that induce the reflexes in the pig, the reflexes in the ants, and other animals that detect the fearful smell, that doesn't seem to happen. The only one that might be reflexive, and who knows, it's, when we're dealing with infants, it's sometimes hard to know what's reflexive, and maybe something that's a little bit more sophisticated is that you do see infants when they are put in a crib that has their mom's odor on a little swab, which you're seeing right here. There's the little cotton swab. The infants, to the degree it can, oh, look at this little guy, will try to crawl towards it. And I just thought maybe that's a reflective response. The infant is crawling towards its mother's smell. Maybe that's like a releaser pheromone from the mother having a reflexive effect on the infant, but it's still unclear. So what Martha and her colleagues and other researchers have suggested is that at least when it comes to humans, we should consider other types of classes of pheromones that might induce more subtle behaviors and more subtle changes when it comes to humans. And these they have named releaser and primer. Oh, I'm sorry. No, releaser and primary. The primer are the ones we've already discussed, right? Releaser, the reflexive one. Primer is the one that induces the menstrual entrainment. What they suggest is adding some new classes when it comes to humans. One is called a signaler pheromone. And what a signaler pheromone does is something we've already discussed, that it's a smell that communicates information about the donor, about the person who the smell sample is taken from. For example, it might give the person smelling that odor information about whether they're related to you, or whether it's a male or female, or, as we'll see, their reproductive potential. We'll talk about this in just a second, but we have talked about some of them already. So these T-shirt tests that we talked about when we first started discussing smell, where a kid or an adult is given 10 T-shirts and told to pick out based on smell which one is from a sibling, or which one is from a parent, or which one's from a child, a biological child. And people can do that, and they can do it well, and especially if it's a biological, fully biological relative, they can do it very well. And what some scent researchers are saying, that is like a pheromone, right? You're smelling the natural scent that is conveying information about who that person is, whether kin means here a relative. Gender would be, that's something else I guess I haven't mentioned, whether something else that can be done pretty easily. You can smell T-shirts and say 5 of these are men, 5 of these are women. Can you tell which is which? And people can do that well. And reproductive potential, we will talk about a little bit. We've already mentioned it a bit when it comes to natural odor and what we call socio-sexual influences. Really, I remember that term. It was a term we used a few slides ago, where we started talking about the basis of a natural scent being something in the genetic code. Remember this, the major histocompatibility complex, the MHC, right? We'll talk about that in some detail in just a second. But you already know a little bit about what's meant by signaler pheromone. It's something that gives information about the donor. In this case, whether they're related to you, whether it's, your brother or sister, those sorts of things. Okay, we'll get into some more detail on that in a second. The other type that has been proposed, besides releaser, primer, and signaler, is modulator. And these are going to be the sort of influences that aren't going to induce a reflexive behavior in a person, but could have a subtle influence over mood and emotion. Okay, so these would be like a releaser pheromone, but they don't induce a strong response. They're much more subtle. And clearly what I'm talking about here are the fear-inducing odors that we've already discussed, right? So that odor does, kind of change very subtly something about the person who smells that odor, right? Unconsciously, all this is unconscious, remember, right? They're being presented the odor. However, it doesn't make them run for the hills when they smell the smell of a scared person. In fact, they don't even know, they have no conscious awareness they're smelling something of a scared person, and probably aren't aware they're smelling any sort of odor from a person at all. But it does have this small effect. It induces these very subtle changes where the eye blink response is a little bit greater. Faces are interpreted as a tiny bit more negative, and people become a little bit more attentive on a test. These are so subtle, but they're real. And what these researchers suggest is, well, we need to acknowledge them. Yeah, they're not like the sort of things we see in animals, but they are measurable, even if they are so much more subtle than the flight response you see. Right? They are so much more subtle. So let's call those something too. Let's call those modulator. Okay. So now when it comes to humans, we have some new classes of pheromones we can discuss. And let's now see if we can add 2 examples of those. So let's go to signaling. These are going to be, again, the idea that pheromones can convey information about a person. Okay. And so these are possible signaler pheromone effects with humans. So let's go and talk about some detail about MHC compatibility. Remember, when it comes to the compatibility issue with MHC, it has to do with how the immune systems between 2 people, or animals for that matter, can be thought of as complementary. So here's the thing. Let's first step back and talk about MHC compatibility when it comes to animals. You probably know this. We might have talked a little bit about this with regard to dogs and purebred dogs versus mutt dogs, right? I've had both. I used to have a semi-purebred. I think he was a purebred Cocker Spaniel. Supposedly he had his what was it called, AKG papers handy somewhere, but we had to pay extra for those and all that, so we didn't do that. But he was a purebred, and now I have a mutt, Max the mutt, who I still will bring, maybe right after the test, I'll introduce you to. And if you've had purebreds versus mutts before, you might know this too, that mutts tend to be healthier. Purebreds have problems, right? German Shepherds, you might know, have hip problems. My poor Cocker Spaniel had serious weight problems and liver problems. And, this is a known thing. If you ask a vet, they'll tell you, yeah, purebreds tend to have more health problems. Why is that? Why are mutts healthier? Well, mutts have more genetic variability. And that genetic variability is a healthy thing, because what it does is it allows the offspring of, say, a Cocker Spaniel and a poodle falling in love Their offspring, because they are not going to have purebred offspring, are going to have kind of a complementary way of fitting, of their genetics are going to have a complementary way of fitting together. So their offspring benefits from that. Purebreds don't have that much variability. So yeah, mutts are healthier. And that also works in other animals as well. And to some degree, it will work in humans, it would seem. Although there's some controversy of that, but certainly we do know that when it comes to things like an immune system or an autoimmune system, animals are looking for mates that have autoimmune and immune systems that complement their own. Let's make sure we understand the idea of complementarity. Okay, so here's my Cocker Spaniel's autoimmune system, and here's a poodle's autoimmune system, right? If they mate, their autoimmune systems are going to fill in each other's spaces. Their immune or autoimmune systems will fill in each other's spaces. If it's a Cocker Spaniel purebred, and here's another Cocker Spaniel purebred, okay, there's going to be a lot of kind of redundancy there, and the little spaces between that will not be filled in. So genetic variability allows these things to fill in kind of the deficiencies. So, you know, Cocker Spaniel has a good immune response to a certain type of cold, but not another. So the holes between my fingers is what you should think about, right? So it would make sense for the Cocker Spaniel to look for another animal that can fill in those holes so the offspring are healthier. This is what's meant by complementarity, when the immune systems fill in each other's spaces. They fill in what the other immune system is lacking. So that's a good, strong autoimmune or immune system. This is a less strong autoimmune system. Cool. We know this is the case. We know this is the case in mice, in birds, in fish, and lizards. Okay, so you look to see, mice couples, and then you look to see their autoimmune systems and say, okay, here's a mommy and daddy mouse in the natural world. They've just had little babies, little baby mice. Let's look at their immune systems. And you look at their immune systems and say, yeah, they are nice complementary immune systems. That's a good thing for the offspring. They're going to be, sick less often and be more resistant to diseases and that sort of thing. It looks like animals are attracted to mates that have genetics and more specifically immune systems that fill in the gaps that are missing from their own. They have complementary immune systems. How do they do that? It's thought. With animals, they're doing that through smell. That an animal's scent signature is telling the receiver whether or not they're going to have an immune system that kind of fills in the gaps of my own. Here I am, Rosenblum the mouse, looking for love, looking for a mate, and I smell around and say, okay, this is all unconscious for mice, I'm sure too, but that's a pretty smelling little mouse over there. And deep down, the genetics know that this is going to be a good mouse to have babies with, because boy, those little baby mice are going to be very healthy because she's filling in the gaps in my immune system, and vice versa. The genetics are going to make the babies healthy. The question is, does this affect humans in any way? Right, so let me stop here and say, if you go online, there are websites that you can pay to have you and your mates genetically evaluated to see if you have complementary immune systems. Don't do this, okay? It's not worth the money. And it seems that, at least for humans, we do base mate selection on a lot of other things as well, right? Mice and birds, fish, lizards, there's a lot of drive from that, that immune system complementarity that is signaled by odor. But what about humans? Okay, what about MHC in humans? Well, here's what we know. If in fact you have, I might have mentioned this quickly a few slides ago, if in fact you take 10 t-shirts from men, all non-relatives, all strangers, to the women who are going to be the subjects in these experiments, and have women smell t-shirts and rate attractiveness of the different smells from the 10 t-shirts. They will very often select good smelling t-shirts, identify t-shirts as pleasant smelling, if in fact that person's MHC is complementary to their own. So when it comes to, I guess, the aesthetics, the rating of a good smelling scent signature, it seems that MHC might have some influence over it. Okay, all right. So that's kind of interesting, right? So, MHC seems to be driving, MHC and how it's conveyed through the scent signature seems to be driving mate selection when it comes to other animals. When it comes to at least giving a rating of pleasantness on a t-shirt, a woman will choose a t-shirt from a man who has a complementary MHC to their own. There's a lot of overlap, right? You're both Cocker Spaniels, whatever. There's a lot of overlap, and they'll think it's not as good smelling. They like an MHC that complements their own based on scent signature. That, okay, is probably an example of a signaler pheromone. Now, so you're thinking, oh boy, I wonder if my significant other has a scent signature that signals an MHC that is complementary to my own. Well, the truth is, and when it comes to mate selection with humans, a lot more goes into it than just a person's smell. Okay? So while we see in mice, birds, fish, and lizards, partners, okay, that have good, solid complementary immune systems, it seems that we don't see that sort of very clear-cut pattern in human coupling. Okay? There's a lot of other things that go into it. Whether you're both Swifties, for example. I'm kidding, but that a lot goes into mate selection. But it's interesting that at least when it comes to, the pleasantness of a natural scent signature in that context, that does have some influence over it. One other thing, there's some other interesting research. I think you'll find this interesting. It's tiny bit controversial, but I think the research is pretty solid on this. This is now, I should be very careful how I don't want to overgeneralize these results. These are college-age individuals who are in romantic relationships. So freshmen, sophomores who've been in romantic relationships for a few months, heterosexual. The two partners come into the experiment, they get their saliva swabbed, and genetic tests are done on them to determine what their MHC compatibility or complementarity is like. Okay? Is this a couple who have a complementary MHC, a complementary immune system? If they do have a complementary immune system, that is one thing. But if it is not complementary, in other words, there's a lot of kind of redundancy, which leaves spaces in the genetics, which means you might have offspring that are somewhat less healthy. What you find is that women in relationships that are in with a person that has a non-complementary MHC will in fact report having more extra relationship affairs and more fantasies about other men. Okay, I take a deep breath here because I want to be very careful. This is not without controversy, these experiments. I believe they have been replicated, but I think it's worth considering nonetheless. That doesn't, these are not marriages necessarily or anything like. These are early romantic relationships with people in their early 20s. And what you find is, to some small degree, the relationship between the MHC, okay, of the male and female in a couple will predict the number of extra relationship affairs a woman is likely to have, and if they're not having affairs, their fantasy life. How much will they fantasize about other men? So, these were all done with questionnaires and all done anonymously, and no one got in trouble. Didn't break up anybody or anything like that. It sounds, a little, kind of a risky research project, but yeah, it's all done very carefully. So again, if the woman had an MHC relationship with her partner that still left a lot of gaps, okay, so there's a lot of overlap, a lot of redundancy, didn't fill in the holes, then she is more likely to have affairs or more likely to have fantasies. If, on the other hand, it does have this cool complementarity, okay, it complements each other, she's less likely to have extra relationship affairs and fantasies. That effect seems to increase when they are tested, and what I mean tested here is when they are asked these questions during their ovulatory period. Okay? So, if they come in and they happen to be ovulating and they're asked about fantasies, the fantasies do seem to increase. The fantasies about other men do seem to increase during ovulation. Okay? So the question is, does this have to do with odor? And I would say at this point, the jury is still out. It does seem to replicate. It's not like there's no evidence that you're gonna get a divorce or anything like that if you don't have a complementary immune system with your partner. Nothing like that. Nothing like you should choose your life partner based on that, right? We're not arguing that at all. But there does seem to be, maybe it's an evolutionary kind of holdover from these other animals that do seem to take that into account when it comes to mate selection. They do seem to take into account to a large degree what the complementary relationship is between another animal's immune system. Okay. Let me just step back here and make sure we all understand what I mean. I'm saying this a lot, complementarity. So this is if there's redundancy between, remember me, I'm doing this here. You can see this good, right? What this means is that the immune systems between two people are too much alike, and it's leaving gaps so that the offspring will have gaps in their immune system and not be that healthy, not be that resistant to diseases. If two animals find mates, though, that have a complementary immune system like this complementary MHC, right? Then they will have strong offspring that will be very resistant to disease. And it's thought that the nature of that complementarity, okay, is signaled by the scent signature of the animals involved. It's thought that animals certainly find mates this way. At least this is one of the criteria, and it's an important criteria. For humans, we don't find that people are less likely to stay married or anything like that, but you do see these little holdovers, these what I'm calling evolutionary holdovers, where young women will have more fantasies if they're with a man that doesn't have that kind of complementarity. Possibly that is signaled through scent signature, as we know it is for animals. Okay, let's go on and talk about other things that is signaled, possibly through smell. So these will all be further examples of of signaler type of pheromones. Any questions about what I just went through, though? Because sometimes people go, "Oh boy, I better test my partner's scent signature or their MHC compatibility." don't take that. I mean, it's a known thing. It's been, kind of supported by the research, which has led folks to start little companies where they'll tell you what your chances of your relationship working out based on your genetics. There's so many things. We're so much more sophisticated than that, and you probably know that. So it's not worth it. Okay, but let's talk about another type of similar pheromone. And this has to do with symmetry. So we don't talk about this too much with humans, but we do talk about it with animals. That body symmetry, okay, can tell you something about the genetic health. So we're not talking about the matching genetic relationship between two people. We're talking about, or between two animals, we're talking about the general genetic health of a specific animal. Okay. If you've ever raised animals, if you've ever worked on a farm, anything like this, you might know this, right? So sometimes animals, after they're born, show that they don't have very strong body symmetry. And if you look this up online, you'll find pictures of horses that have kind of misshapen hooves. They have two hooves that are the a different structure, that type of thing. And what we're talking about here with symmetry, by the way, is typically vertical symmetry. Samuel is asking a question. Let me just finish this sentence and I'll get right to it. So when we're talking about body symmetry, we're talking about symmetry this way. Okay. Does the left side of the body match the right side? And for some horses, or other farm animals, for example, I'm sure animals generally, they are less matching across the two sides, and that's what this is trying to show here, that the hooves don't match. So this would be an animal that we would say has low symmetry. Okay, we'll talk about the ramifications of that in just a second, but let me answer Samuel's question. So what is the pheromone that correlates to the wandering mind? I don't know. I've not heard of pheromones that are related to a wandering mind being, having fantasies. If that's what you mean by wandering mind, I don't think that anyone has established the actual chemical composition. We're talking about if it is communicated through scent signature, then it's going to be, the natural scent of a person is going to communicate, something about their immune system, something about the structure of their MHC, and that could possibly have an influence over women if there is too much overlap and therefore not enough kind of complementarity. Yes, what the question you also asked, is this just true when a woman's ovulating? And no, it is not just true when a woman's ovulating. She will say she has, possibly not all women, but some women will say they have more fantasies if in fact they have a man who has an overlapping and not kind of supporting MHC. But it turns out they will have more fantasies. That difference will increase when she's ovulating. So it's not just with ovulation; it's just more pronounced during ovulation. Okay, but they don't know. I mean, they don't know the chemical. So when there's low compatibility with the smell, there's more fantasies. Exactly right. But I want to be careful here. What I'm saying is when there's low compatibility in the genetics, okay, when it comes to, again, the MHC and immune and autoimmune systems, there are more fantasies. It's not absolutely known if that information is conveyed through smell. It's assumed it is because it's true of other animals. Does that make sense? So what we can say is, that we can say that the genetics can predict the relationship between the genetics, whether it's too redundant or it's nice and complementary. Okay, that will predict the fantasies, and it's thought probably pretty likely that that information is signaled through smell. It's just that's not been established through the experiments, the genetic thing he has. Okay, great. Glad that works out. Let's go back to body symmetry. It's interesting because body symmetry. So if you're raising animals, this is something that farmers, dog breeders know. If they have a new animal, a baby animal that has low body symmetry, they will know. Well, we probably don't want to breed this animal because they are not going to be as healthy. They will have slower growth, reduced longevity, fertility. So they will know that for whatever reason, the animals that have low body symmetry are not going to be as healthy, and they won't have as many offspring, and their offspring will not be as healthy as animals that have more body symmetry. So again, we're talking about hooves, we're talking about antlers, we're talking about maybe shoulder musculature. There's a lot of things you can look at in animals. For the most part, most animals are pretty symmetric looking. If you look on both sides, right? If you look at your doggy right now, you'll see that they look pretty symmetric. They're probably healthy and all that, right? But sometimes animals are born without that. So it's interesting because that symmetry is something other animals can see, right? And potentially other animals find less attractive or are less likely to mate with that animal. The question is this also true with humans? So a lot of the research on symmetry when it comes to humans is done with faces, which is interesting. And that's why I have this poor gentleman right here. If you were to draw a line down the middle of his face, you would see that, it looks like one side of his face is maybe drooping a little bit more than the other. There's this side of his face is maybe down a little bit more from that. We would say that this face has less symmetry. Now, let's all be careful because I know you're thinking of running to the mirror. We're talking about major differences here. Everyone has some asymmetry on their faces. Like we have moles on one side, not the other side. Our smiles are a tiny bit asymmetric. That's fine. We're not talking about those sort of very subtle things. We're talking about more major situation. And it's interesting, when I wanted an image for this slide, I said, okay, let me look up how do I get a picture of a face that's asymmetric, and you find a lot of asymmetric faces in mugshots from police stations and prisons and places like that. You find faces that are more asymmetric with folks that have gotten in trouble potentially. Which is interesting. And there is research that when it comes to major asymmetries in faces, that you find that those people generally have somewhat lower IQ. They often have worse physical health. They have to deal with more health issues, more immune problems, that sort of thing. And people that have asymmetric faces have somewhat worse mental health as well. Again, don't worry about your face. It's perfectly fine. I understand people want to say, oh, maybe I'm one of these people. You're not. You're in a good college. Yeah, okay, so Samuel brings up this very good point. Yeah, we're not. We're talking about a genetic basis of asymmetry. Where, all of us have had scars on one side of our face and not others when we were kids, or we got little scars or burns or something like that. We're not talking about asymmetries based on some sort of event a person had in their life. We're talking about asymmetries that a person is born with that have a genetic basis. And when those asymmetries exist, you do see somewhat lower IQs and depressed physical health and mental health as well. When a person has higher body symmetry in humans, it's just the opposite, right? They have somewhat, they don't have the problem of lower IQ or worse physical and mental health. And you find people that have high body symmetry and high facial symmetry are, especially men, are likely to have more sexual partners. And research suggests is they can actually induce more what's called copulatory orgasms through standard intercourse. They can induce more like that. Now, who knows what the relationship between these things are? The relationships are between these things. But what we're interested in on this slide is, in fact, the relationship between symmetry, body facial symmetry in humans, especially human men in this case, and smell. So let's go back to our t-shirts again, our t-shirts experiments. We're going to have women smell the 10 t-shirts of stranger men, okay, men they don't know, they've never met, they have no relationship to, and have them rate them in order from 1 to 10. And then we'll take a look at what the basis of that might be. You already know one of the bases, and that's the complementarity between their MHC and the donor's MHC. But there's something else that plays a role, and it has to do with the symmetry. So what you could do is you could, get, maybe all folks that are in prison. Okay, I mean, there's asymmetries in the non-prison population, of course, but 5 t-shirts from guys that have asymmetric faces, and 5 t-shirts from guys that have very symmetric faces, and ask women to smell and to see which smells of the t-shirts they like more. And what they find is, and this is actually something that's very clearly determined, or women seem more sensitive to this difference between symmetry and smell when they're in their fertile phase. I don't think this particular one will work if a woman is not in her fertile phase, but if she's, she's ovulating and she's asked to come into an experiment, smell t-shirts, she will like the smell of a symmetric man's t-shirt more than an asymmetric man, okay? Which is interesting. So, again, she might fall in love with a person who has an asymmetric face because there are so many other things that go into it, However, it's interesting that it seems that we do know that animals that are asymmetric, okay, that have an asymmetric body, are less likely to have many offspring, okay, and have many mates. Right? More symmetric animals will have more. And it's thought that what's going on here is the selection of genetic health. That, yes, it's something you can see, right? The genetic health of a person, whether you know it's genetic health, might be conveyed in the visible characteristics of a person or an animal, but it also might be conveyed in their smell. Okay? So what you're doing here, when a woman smells a series of t-shirts and likes the symmetric man's t-shirts better, like that smell better, what's going on here is they are essentially smelling the genetic health of that man. If a person is genetically healthy, they will have they'll be more likely to have a symmetric body and face and have a more pleasant scent signature. If they are genetically less healthy, they're more likely to have an asymmetric body and face and have a less pleasant scent signature. Okay? So there's kind of this dual way, which we'll talk about later. It's interesting. It's one of these multi-sensory kind of signals about genetic health. One has to do with what you can see, asymmetric face, and another has to do with what you can smell, unpleasant odor, unpleasant scent signature. So genetic health is conveyed to both the eyes and the nose in this sense, right? So it's a signaler. Again, this is the sort of thing that Martha McClintock, her researchers have suggested is an example of a pheromone that's communicating something about the donor, that this is a more healthy person genetically, a less healthy person genetically. And it does seem to heighten a woman's sensitivity when she's in her ovulatory phase. A couple of other things here I should mention about sense signature that you might find interesting, that I think also are examples or could be taken as examples of a signaler pheromone. And that is when a woman's in her fertile period, she not only likes a man that is genetically healthy and therefore symmetric and has a immune system that complements her own, that fits well with her own, she also likes the smell of more dominant men. Now, there's lots of ways of evaluating this, and I actually should go back and check to see if this is still the case. But there are ways of using questionnaires to determine if a man is more dominant in social situations, whether they're likely to be bosses, leaders in say a military context, that sort of thing. And at the time I wrote the book, at least, there was research showing that when it comes to smelling t-shirts, that's something else that maybe can be detected, is that a man that has been established to be in a more dominant role has a more pleasant smelling t-shirt, a more pleasant smelling scent signature. That is something that is detected mostly when the woman is in her fertile period. If she is not in her fertile period, that's something that she might not be able to detect. So these last few experiments that I've talked about are using women in her fertile period. And what that's certainly showing is that women in their fertile periods do seem to be more sensitive to these things. So again, another type of signaler pheromone, signaling not just MHC compatibility and genetic health, but also possibly signaling the potential dominance or the potential, I guess dominance is the right word for men. I should go back and make sure that stands though. But let's keep it on. Let's assume it's okay and it will be tested. But I think I'll go back and check that just to make sure. So far, though, when it comes to these sort of signaler pheromones, we mostly talked about women detecting the smell of men and those characteristics as they are conveyed in men. So most of the signalers are coming from men to women, often when the woman is in her fertile period. But there's also some examples in the research showing that women signal things to men too through scent. So now you put men in an experiment where they're smelling t-shirts that have been worn by women. Half of these women when they wore the t-shirts were during their fertile periods, and half of the women, 5 of them, were worn by women who were not during their fertile periods. And in fact, when it comes to smelling the t-shirts, the men will like the natural scent signatures of the fertile women better. They'll like other things, other things do play a role. We do know the MHC thing does play a role here too. I don't know if that's the symmetry and the dominance thing has played a role, but we do know that, when it comes to smell and men smelling women, they do like the t-shirts worn by ovulating women better than a woman not in that part of her cycle, which is interesting because and that's why I have this little monkey here. We do know that fertility is something that is often very conspicuously signaled in other animals, right? Here's a monkey saying, it's time to ovulate, look at my butt, right? That's a very conspicuous sort of signal. Other animals, birds have different plumages, all that sort of stuff. We always kind of assumed that when it came to humans, it was not something that was advertised, and there's reasons that might not be advertised evolutionarily, right? But it seems that maybe there is some little subtle advertising going on, in the sense that men seem to like the smell of a fertile woman a little bit better, right? So maybe, fertility is not concealed in this sense. All right, let's just kind of do a quick summary of everything we've talked about on this slide. We are looking at a type of pheromone here that is signaling information about the scent that a person is releasing. And we've talked about other things with regard to this, like kin selection, that sort of stuff. But look at this list here. It seems that at least the research shows there are contexts in which a woman can detect all these aspects of a male donor, can detect the potential MHC compatibility, can detect the genetic health through the whatever bears not just on the scent signature, but also has an influence on symmetry, body and facial symmetry, and potentially dominance. All of these would be examples of what Martha McClintock and her colleagues are calling signaler pheromones. They signal something through scent to a receiver, somebody who's smelling it. All this stuff is done unconsciously, right? Although, I mean, some of the experiments are asking people to smell the t-shirt, certainly. But any influence on mate selection is likely done unconsciously. And it's also, let's be very careful here, it's also clear that this is not the only thing that influences mate selection with humans. It's an important thing to say, right? Don't go waste your money on genetic testing with you and your significant other. It's not very predictive of longevity of relationships and all that. But the fact that it still seems kind of even maybe a small part of the equation, is interesting, as it's known to be a large part of the equation for other animals. Okay, we're now going to go on to the other type of pheromones that Martha McClintock and her colleagues have wanted to add, and that's modulator pheromones. Now remember, modulator pheromones are kind of like, whatchamacallit, what I call a reflexor, hold on a second, the releaser pheromones, sorry. So the releaser pheromones, remember what they make you, they do for animals. It makes you run for the hills if you're a mouse. It makes you go into the mating stance if you're a pig. Nothing like that for humans, but that doesn't mean there aren't subtle influences. And when those subtle influences come from another person's scent, they're categorized as modular pheromones. So what are some examples? We've continued to talk about one, the eye blink response, right? That is kind of a nice example of what would be a modulator pheromone. That fearful smell does not make you run for your life. It doesn't make you run out of the lab, right? What it does is it makes you blink a little bit faster, a little bit harder. If there's a loud sound, the loud sound induces the blink response. The smell just changes it and modulates it a little bit. And it has these other very small effects too on how we interpret facial expression and tension on an exam or something like that. Very subtle effects. Very, very subtle effects. What about the other type? What about the type that kind of puts the female pig in its mating stance? Are there influences like that? There are fearful influences, but they're very subtle. Are there influences that have any sort of romantic influence at all when it comes to humans? Because as we started our discussion, right, you can buy pheromones. Oh yeah, I thought of you. I thought of the whole class. I thought about this topic when I was watching Shark Tank a few weeks ago with my kids. We love Shark Tank. We think it's really fun. But someone was trying to sell a pheromone-based shampoo. And the folks that were making the proposal said, we know that pheromones can make people attracted to each other and all this other stuff. And as he often does, Mark Cuban always plays devil's advocate when it comes to these sorts of claims. And he said, okay, if your product is resting on the idea that pheromones are going to, induce romantic feelings or arousal or something like that, you're going to have to show us evidence that not only does this happen systematically with pheromones in general, which he said, I don't think there's that evidence. And you're gonna have to show us your product does this. And they said, no, we haven't tested with our product. But we do know that the research, and before they could finish their sentence, Mark Cuban said, I'm out. I actually love when he does this. As a scientist, he's, I love when he kind of pushes the claims, whether it's a health food claim or, our smoothie mix will lower your cholesterol. And he said, do you have evidence of this, that it actually does it? And they'll say, no, but we do know that this type of thing can I'm very glad he does that. He's very cautious when it comes to the scientific claims made by folks who don't do the scientific research. This is a good example of this. But is there any evidence that something like a pheromone can have an influence over somebody else when it comes to, romantic feelings, when it comes to something? And there's a little bit, but it's so tiny. Don't go buy your pheromone shampoo. Okay. What I said here is putative, and I don't know if you're familiar with that word, but that means what the researchers did in this experiment is they kind of put together a chemical based on the secretions of men's, what men secrete, that is thought, if there's going to be any sort of pheromone that has any sort of modulator effects on mood or arousal, it could be this. So the only reason it says putative is that it's not actually, they didn't take the pheromone from the men, they kind of re-synthesized it. I just wanted to be cautious there. That's why it says that. So again, all these experiments involve undetected. So, the women might be wearing a cannula, but there's a cover story where it says that we're going to be testing your oxygen levels, so you need to wear this. And what they're actually doing is they're pumping this scent in there, but it's, again, too low to be detected. It's in there, but it's much too low to be detected. They don't smell a thing. And what it will do, if it is being pumped, it will have very subtle influences on, we'll talk about moods at the end, but heart rate will increase a tiny bit. Skin moisture, which is detected by what's called the galvanic skin response. Don't worry about the technical details of that at all. But if you're in a lie detector test, like you've seen them on TV and movies or whatever, they put something on your finger and that tests your skin sweat. That's how this is tested. And yes, women will sweat a tiny bit more. It's not visible. It's not even something you could really feel on the skin, but this device will test very subtle changes in skin moisture. And cortisol levels are tested through swabs in the mouth, and all those things will increase a tiny bit when a woman is exposed to the undetectable scent of a man that's created in the laboratory. It also will elevate mood a tiny bit. So they will do a questionnaire and say, right now, how comfortable do you feel? How happy do you feel? How satisfied do you feel on a scale from 1 to 7? If they don't have exposure to that putative pheromone, if they don't have exposure to that smell, they might say on a scale from 1 to 7, they're about a 5, happy-wise. If they do have exposure, they might say 6, but not all women will show it either. And as soon as that smell is shut off, if they're asked again, it would probably be 5, right? So these are all very, very subtle and very temporary effects. The smell is shut off, cortisol level drops back to where it was, same with skin moisture and heart rate and mood. So you do see a little bit of an elevation, but it's very short-lived and it's very subtle, and it's not necessarily even detectable across all women. statistically, if you look at a lot of women, 100 women, you will see an overall average that just raises a tiny bit. Minor changes. Also, another experiment, you can ask the women to watch an arousing clip, video, an arousing video, arousing film clip. So, I don't know, what's an arousing, what do women watch now? Okay, 50 Shades of Grey, whatever, is that the right name of it? I don't know. Oh, okay. Samuel has another question. We'll get back to that. Sorry about that. I missed it before. Say you're watching 50. Say that's your type of movie. So you kind of dig that, and how the romantic scenes are depicted, and you enjoy it. It makes you a little aroused, and you're watching the scenes, and you have a cannula on, and you are being presented this pheromone again. This putative pheromone, right? If in fact you are then asked at the end to rate your arousal, how aroused were you by those scenes? You might rate your arousal as a little bit higher than if you didn't have exposure to that putative pheromone. Again, on a scale from 1 to 7, you might say, oh, I would rate my arousal at a 6. And if you weren't being exposed to that putative pheromone, you might say a 5. We're talking about a tiny shift. But testing across 100 different women, you would see a tiny bit of an elevation like that. Okay, let's go to Samuel's question. Any studies that target a genetic signature? Scent signature, I think that in terms of the scent signature, it's thought to very clearly have a genetic basis, if that's what you're asking. And we talked about some of the experiments early on where like identical twins don't have the genetic difference, and therefore don't seem to have a difference in their scent signature, at least in terms of how parents have difficulty recognizing T-shirts across the identical twins, and same with tracking dogs, if that's what you mean, Samuel. So yes, it's thought that the scent signature has a very strong genetic component. If you're asking something else, go ahead and type more.
Okay. Hello?
Yes. Yeah, go ahead.
Yeah, so what I mean is that there's a complement that is there any studies where they kind of might like chemically produce a complement signature and then see if it affects people?
So in other words, what you would do is actually like synthesize a scent signature yeah, that's a really good question. I don't know if they've actually synthesized something like a scent signature. But what we do know is, you have the scent signature by a male that's, worn the T-shirt, and then you look to see if a woman finds that attractive. It's if they do find it attractive, then you can do the genetic testing on both the woman and the man to see if they have complementary immune systems, and they do. So I don't know if there've been any studies that actually synthesize the scent signature, but we know enough that the natural scent signature is predictive of the genetic complementarity between the two people. Does that help? Is that what you were asking? Yeah.
Yeah, thank you.
Okay, sure. Thanks for the question. I appreciate it. But let's go back to the sexual arousal thing, because this is certainly the most controversial, right? This is the thing that they're trying to sell you. They tried to sell on Shark Tank. This is going to make you an irresistible mate when you go out to parties and all that sort of stuff. No, there's no evidence like that at all. It does elevate arousal a tiny bit, but what else does? And that's why you have your friend. I don't know if you guys recognize this right here, who this is. This is Marvin Gaye. And I love him as an example. And in fact, I think the chapter is called Marvin Gaye for Your Nose. That's really what you're talking about. So, I assume you've heard of Marvin Gaye. Maybe your parents listened to him. That was my generation's kind of romantic music. When you wanted to kind of be put in a romantic mood with your significant other, you turn down the lights, maybe light some candles, and you put on Marvin Gaye. It's very romantic, and let's face it, sexy music, and it helps you get in the mood. And that's probably, to the degree there are any scent influences, that's probably the level it's at. Certainly, if you're hanging out with somebody who you really just consider a friend and the Marvin Gaye music goes on and you're not interested, no way. Say, I'm sorry, that's not how I see our relationship. I really just want to be friends. Let's put on some Taylor. Well, maybe you find Taylor Swift, arousing. I don't know, but you won't want Marvin Gaye on and the lights low and the candles lit if you just want to have a friendship with the person, right? It's not going to make you all of a sudden feel romantic towards the person at all. You are not like the female pig and have that reflexive, kind of sort of reaction, right? And that's the way to think of it, probably, for humans, or, we used to call this a bachelor pad. If you've seen the Austin Power movies, you've seen things like this back in the 60s and 70s. Men would sometimes have bedrooms that were sort of, kind of inducive to the kind of romantic kind of, situation where they have maybe a circular bed, or water beds were very big back then, and maybe satin sheets and all those sort of things. And, you could change the lights from a remote control. I mean, it was that nerdy, trust me. It worked like that. And, if you're with a person that you're romantically involved with, maybe it has a little bit of influence. Maybe it puts you in the mood a little bit more, right? But again, if you have no interest, if you had a really bad day, the last thing on your mind, it's not going to induce you to want to be romantic if you're not in the mood. And that's, again, think of the, influence of the male scent like that. It can maybe elevate your arousal if you're already aroused or you're open to arousal, because something else is making you aroused, like a video you're watching or something like that. But it's not going to make you feel aroused from scratch or anything like that, right? It doesn't work like that with humans. Okay? We'll talk about whether it works like that with animals and pigs in just a second. But before we leave this, I wanted to kind of just mention a couple of other things that you might find interesting in the research. And that is that, having a male smell there, if you're a female, is not the only thing that can kind of facilitate more arousal. As it turns out, oops, oh, I took that out. Okay, that's fine. Let's leave it out then. Okay, the book talks about this. Nursing smells. Go ahead and read it in the book, but I think I did take it out of the exam. I'll let you know for certain. But nursing, the smell of another female nursing, is known to enhance sexual fantasies a little bit. And there might be some evolutionary reasons for that have to do with coordinating pregnancies and coordinating nursing and that sort of thing. But it's interesting. It's not a male smell that's doing this. And in fact, it's in one of those experiments that involve an undetectable smell again. But it's not the male smell. In this case, it's a female smell that can do that, and it has to be a female smell that is based on a nursing mom. Why in the world would that enhance arousal? There's possible evolutionary reasons, but again, okay. But let's finish this up. And there's our, what's it called? A trackmate pheromone concentrate. Right? It's an old stuff. I don't even know. These pheromone sellers, they go in and out of business pretty quickly. I don't know if that one's in business, but don't buy it. Let's talk about human pheromones. So what this slide says, the effects of subliminal odors are subtle and complex, and that's especially true when it comes to what we're discussing as, quote, pheromones. Okay, Biologists that study animals would say because you don't see the reflexive stance in humans, because you don't see the fearful sort of reflex, and you don't see other things, we're not going to call these pheromones. But other researchers, and Martha McClintock says, yes, we can call them pheromones, but let's realize that most of them are not releaser pheromones or primer pheromones. Most of them are signaler or modulator, and the signaler ones might give information about genetic health and genetic compatibility. The modulator ones might have some very, very subtle influences over fear and possibly arousal, but they are much more subtle than the sort of things we discussed before with regard to the releaser. There's some evidence for primer. I mean, the primer, the kind of quintessential example of a primer pheromone in humans would be menstrual entrainment, and the idea that menstrual entrainment is based on scent, at least to some degree. But there's very little evidence that there are pheromones that can actually induce some sort of releaser type response in humans. Very little evidence for that. Again, if you're exposed to a fearful smell, if you're exposed to an arousing smell, right, you're not going to react like a female pig that goes into a mating stance or anything like that. Far from it. You're not going to, run out of the lab room if you got a fearful smell. It's not going to induce a reflexive response. And Martha says something very interesting to me, and she says she's written about this before. If you look carefully at the animal research, when it comes to the pig especially, releaser pheromones might not be as, I guess, strong and as determining as they are often portrayed to be. So if you look at the pig research, you can't induce a female mating stance in a female from the male's smell alone. Okay, something else needs to be there for the female to go into a mating stance. So you can't take a sample of a male pig who's 5 rooms down and just bring that smell sample to the female pig and get her to go into that stance. It doesn't work like that. The male has to be in the same room. And the female doesn't necessarily have to see the male. The male can be behind the female. But the male needs to be in the same room, and it might be that the female needs to hear the male in the same room. It needs to know, the female needs to know that it is something that's in proximity. So the smell might have something to do with the copulatory reflex, but it might need something else. It might need sound. It might need some evidence that the male is there. So the idea that just a smell can induce a reflex in a pig probably isn't true. And that also might be the case for the ants and the mice as they run away. There might need to be something else there. So what Martha said is she's not 100% convinced that releaser pheromones are something that, true releaser pheromones are something that exists in the animal world too. It could be that, these pheromones that we're calling modulator have a stronger effect, but it might not be this determinate effect, meaning that no matter what, the animal smells the fearful smell, the animal smells the male smell, and it's a reflex. It might be more complicated than that for animals as well. It's clearly more complicated for people. The pig stance might need, the pigs, for it to go in a stance, might need something more to tell it the animal's there, to tell it the male's there, like the sound. So when it comes to subliminal odors, at least for humans, and this should be the takeaway, is we're moving out of sound and going to be talking about taste in just a second. As we do that, it should be clear that when we're talking about the effects of smell, it's a much more complicated story. Subliminal odors are not determinate, especially when it comes to humans. They can give information, they can change things a tiny bit, but we're talking about tiny changes that are really no more pronounced than what you would see with Marvin Gaye music or low lighting or something like that. Okay. When you see with humans, you might see very subtle changes, but they're not going to make you fall in love with somebody you're not going to fall in love with for other reasons. They're not going to make you so scared that you have to run away. These are very, very subtle effects, but it's interesting that the effects are still detectable. They're still something you can observe in the right laboratory context. It suggests that that's part of our perceptual repertoire that we're unaware of. So it's a nice example in that sense. Okay, cool. Let's take a 10-minute break and come back at 10:42. All right, we are back. Hold on just a second. Let me just fix something here. All right, good. Yeah, everyone will be happy. Okay. All right. So let's talk about taste. So this is, I guess, chapter 5 in the book. Oh, here, let me just do something. That'll be all right. Any questions on anything about the previous material or anything to do with the test? I just heard a rumor, meaning I saw an email, and I think your TAs tomorrow are going to spend some time going through a little review game where, kind of like Jeopardy or something like that, where they test you on questions from the content of the class and, Jeopardy. So that's nice of them to do. So that should help a little bit. Okay, any questions on the test or anything? All right, so let's do taste. So, like smell, I really knew nothing about taste. I teach perception, I do research in perception, but for most perception classes, smell and taste is like a single lecture. You kind of spend most of your time on seeing and hearing and some on touch. So I didn't know very much, and I was kind of excited to learn about the research. But before I started writing those chapters, I found out about this restaurant in LA called Opaque. It used to be on the Sunset Strip, kind of next to the Laugh Factory. I don't know if you're familiar with the Sunset Strip. But the reason I was interested in it is because it advertised as a restaurant that is completely dark on the inside. There's no light whatsoever. I mean, you're sitting in the dark at your table and being served in the dark and eating food in the dark. You have no idea, where the next table is. There's, the decors, all the walls are painted black, and there's curtains to make sure that there's no light whatsoever. Everyone has to have their phone off. And the idea, at least the way it was advertised, was by eating in the dark like this, you'd have a chance to really taste the vividness of your food because you wouldn't be distracted by the sort of things you're usually distracted by. Other people in the restaurant, people walking around, the decor, all that sort of stuff that, we usually are looking at while we're eating. So without that, you can really, really concentrate on your food and taste all the vivid flavors of your food. Sorry, there's garbage trucks on the side. So this is a phenomenon now that's kind of gotten popular. It's called dark restaurants. And I guess they started in Europe and then came to the US. And I think most major cities have at least one of these now. The one I went to, Opaque, which is the one that's going to be portrayed on a video you're about to see, moved, I believe. I think now it's in Santa Monica somewhere. I haven't followed it. But I believe it's still open. Last I looked, while we're all watching the video, I'll look it up to see if I can find out any other information about it. But I will tell you, it is as dark as any place I've ever been in my life, and it was a really interesting experience, especially for someone who's a perceptual psychologist. I went with my wife and another couple, that we're close friends with. I wanted their input too to see what they thought of the experience. But here's a video that will show you a little bit about what it's like.
I came here tonight because it sounded different. They said it's dining in the dark. It's probably a little unique and disturbing, but that's why we're here.
Welcome to Dining in the Dark. Y'all make this special.
Oh, thank you, Mike.
Dining in the Dark, it's a dining experience in a literally like dining room, dark dining room where all the food is served by blind servers, and that just makes you have like a totally deeper, greater understanding of the other senses that we all have but often neglect.
Found my butter knife. Was there any butter?
Yeah, it's in the middle of the table, which is here.
The whole idea of dining in the dark comes from Germany. Ever since that first restaurant got so popular, so trendy, so like the thing to do, they started sprouting all over Europe. I took an American friend and she just said, like, why is the good stuff always happening in Europe? Why do we Americans always have to travel to Europe to actually go do fun stuff? And then we just like started talking about it, and that's how we came up with the idea to do this in the United States.
OPEC dining, the blind leading the blind. What the awesomeness of it is, although they can't see, I can guide them into a great evening of dining.
Tongue it in, either way.
All right. Someone is entrusting me, a person who cannot see, to guide them and lead them into a dining experience.
The server takes them to the table. The servers exactly know where they're going. So the diners, as soon as the door shuts, they're lost. The server really acts as a guide for the entire night. The blind server is like the only person that can actually do the job, because they have like had that disability, which now becomes their ability.
I can't believe, no, you really can't see anything.
Just like you want your eyes to adjust.
No, I know, there's no way. I may as well just close them.
I'm not sure.
So no holes in Vermont?
Yeah.
Okay. Is that my water?
Yeah.
People really go in there and just have such a blast.
Raise your hand.
They just laugh. They just like they behave like kids.
Where are you?
Are you trying to feed me?
Yeah.
You're awesome.
Warm, cold.
That's awesome. At first, when I was walking in, I'm like, I can't do this. I know I can't do this. But then once I got settled, I don't know, it was just crazy. You just like adapt pretty quickly.
There's a complete suspension of self-consciousness. It just brings everything else into sharper focus. I'm on your right, Brett.
When you go to a restaurant, what the experience is obviously the ambiance, you know, to be seen in there. You don't experience that here. It's more really tasting the food and people's conversation.
This is like an experiment at being born. It's genius. I like this a lot.
You're really tasting things. You're really tactile. You're really feeling things. You're really hearing things a little more acutely than you normally would.
Pretty tasty. It's good. It was incredibly hard to cut the steak, and then I noticed after a while, I was like, well, nobody can see me, so I just picked up the hunk of steak and started chewing it like a caveman.
Hey, Michael,
Yes.
would you mind helping us and escorting us out?
Yes, I will. It's almost indescribable unless you're actually, doing it.
Gives your eyes a definite break and allows you to focus on everything else and play with your food, which is always a good thing.
Right.
Coming out, it was like very humbling for me. Like I feel very humble and kind of just appreciative of how easy life really is in a sense for me.
Hey, I'm glad you guys came. All right, my friend. Well, come back again.
We will.
More than welcome.
Thank you so much.
Yeah, Michael's great.
All right. I looked it up. I have more information for you, but I will not tell you yet. I will tell you what my experience was like. It's in some detail in the book. But when we got there was somebody sitting outside in the lobby. The lobby's, a perfectly lit lobby. But there was somebody sitting there, and they had a glass of wine, and they were shaking. And I guess that happens every night. At least one person comes out because it's such a kind of strange experience. This person ended up going back in, but it took a little while for them to feel comfortable. It is a very strange experience. But the reason why there's a place where you can actually sit, well, probably one of the reasons is for folks that aren't comfortable. Another reason is that's where you order your food, because you can read the menu there, and you're given 3 choices of how to order your food. You can do it, normally, where like you go through the menu and you pick all the courses and as you normally would in a regular restaurant. And that's what my wife and our friends did. But I decided to be a little adventurous and just like kind of pick a category. And I just picked vegetarian. I'm not a vegetarian, but I just thought maybe it would be safer. So I picked that, and then I left everything else up to the chef. You also can choose to be completely surprised if you want, which I was too scared to do. But anyway, you go in, and it is really dark. I've never been in a more dark place in my life. It was the sort of thing where you're not sure your eyes are opened. It's so dark in there. You see absolutely no light at all. Any direction you look, it's completely dark. You're just kind of spending a lot of time talking to your friends, so you know where everybody is. We're at I guess a normal table where people are sitting. We're sitting across from our friends and all that. I had a beer, and then I think we had probably a total of, probably between us we had, I don't know, we all had appetizers and a main dish, and all had a dessert. So maybe we had, I don't know, 15 to 20 different things that we were trying. The question was, of course, how to, can we taste our food better in the dark? That's what it was advertised as. We didn't know what to think, but I'll tell you that. I'll tell you what the answer is in just a second. But without knowing what things were, you saw kind of some funny things happen there where you don't know necessarily what what the vegetables are and that sort of thing. And there's a lot of situations where you miss your mouth with the fork and it goes into your cheek, and you're chewing on something, you're not sure what it is. At one point, I was chewing what turned out to be a carrot, but I had no idea what it was. And it was kind of like, it was kind of really chewy, and it was like the size and shape of like a finger. And I thought, this is really strange. I thought, oh, maybe the chef was blind too or something. No, I'm kidding. But it was a little strange, but yeah, I'm glad we did it. I'm really glad we did it. And it was an interesting experience. Let me go to the next slide, and I'll tell you some other information now about it. Of course, what we're interested in is, in dark restaurants, does the food taste better? Okay, does the food taste better? And so I'm not going to tell you yet. What I'd like to tell you instead is the research. So what does the research suggest? Well, the research suggests something interesting, and I didn't know this. I didn't know this until after I ate at the restaurant. And I guess maybe I'm not completely surprised about this, but the research suggests that what you see influences what you taste in a big, big way. So, for example, a lot of the research on taste, of course, comes from companies who are testing their products and trying different things with their products and trying to determine what they need to do to change not just the flavor of the products, but also the view of the products. And you folks probably know that a lot of what you see influences how things appeal to you. I mean, the cooking shows you watch. My wife watches all of the Great British Bake Off. She loves it. and my kids like watching the kind of more kind of normal cooking competition shows where it's Gordon Ramsay type shows, that sort of thing. And I mean, they're really successful as a whole network based on this. And why do we find these so interesting, and why do we find it appealing? And that's because we know that the sight of food has an influence over how things taste. And certainly the research shows this, that fruit drinks can depend, the actual taste that people report tasting can depend very much on the color. So these are like the things that you guys might know would be like Gatorades. In the old days we had like a lot of sugary drinks like Kool-Aid, and what else did they have? High C, things like that, where they'd be sugar drinks that were different colors. But this is also true of the Gatorades and other sports drinks. If you have a chemically composed drink to taste like it's orange, but you've put red food coloring in it, people will then sip it and often say it has a cherry flavor, and vice versa. You can have all the flavor components of a cherry drink, but change its color to orange, and people will often say it's an orange flavor. What they experience tasting will depend very strongly on what they're seeing the drink look like. So yeah, you can swap flavors that way. That's what the marketing and kind of food production research suggests. The other thing is, and this is going to be interesting with regard to the dark restaurant question, the intensity of the drink will depend to some degree on how bright the color is. So, certainly for the sports drinks, they're going to make the colors look brighter, so the flavors taste more vivid, where the chemical composition of what's actually affecting your tongue is not changed at all, but the color change makes your brain change its evaluation of the product. It makes a drink seem more flavorful if the color is brighter. A brighter red will make a cherry drink taste more cherry. An orange drink tastes more orange, and that's been well known. So dilute things that don't have a lot of visual appeal don't have very vivid flavors for people. So again, what you're seeing is influencing what you're tasting. And this has been known for a long time with other things. So like cookies, custards, cakes. Custards, I think, are really an interesting example. A lot of these experiments are done with desserts or sweet drinks, or things that are not savory. People usually find these much easier to deal with in experiments, I can imagine. So the custard thing is, a lot of research has been done with custards, because there you can manipulate the texture, where you could look at a custard, and if it has a visibly thick texture, that's going to influence how it tastes to you. And they do some really interesting, clever little experiments with this. So what they'll do is they'll have you taste the custard through a straw. So imagine there's a bowl that just looks just like that, but they put a straw down in it, and then they have you taste the custard and give a rating of how sweet it is, how flavorful it is. And what they'll do is, this will actually be like a magic bowl, a secret bowl, where the straw is actually going to a part of the bowl that is different from what you're seeing. So you'll actually be ingesting a custard that is different from the one you see. And you could be ingesting, say you have two bowls there, and the straw actually goes to the exact same custard, but if on top this one looks thicker and the other bowl looks thinner, okay, you'll say that this one is not as sweet as the other one. Its visible thickness will affect how sweet and how flavorful it is. Typically, if you're interested, the thicker it looks, the more sugar needs to be added for it to seem as sweet, even though, as I just mentioned, the custard isn't changing at all. You're actually sipping the same custard. The straw is going to the exact same custard in both cases. It actually makes it also easier to discriminate between two custards, if that's the case, right? If there's subtle differences, if it in the actual flavor, the visible texture will make it seem different. So if a company is selling two custards, I don't know, the Jello brand sell custards, or maybe you have a favorite organic custard from Whole Foods. If they have more than one type of custard, if they're thin and thick, there might be a difference in how it actually is chemically composed for your tongue, but the difference probably is even more dramatic in how it looks, because that's going to have an influence on your tongue, and that can make the change more cost effective or something like that. Importantly, these changes from what you're seeing are not just, we call these true perceptual changes. What I mean by that is, in these sorts of experiments, we have to make sure that they're, it's not, we're not, the subjects, the consumer isn't actually kind of telling the experimenter what they want to hear or something like that, that there's actually a change in how the perceptual experience works, right? Because you can imagine, if you're evaluating a food, if you're evaluating one of these drinks, or sports drinks or something like that, there's a lot of things that can play a role in how you evaluate it, including the context of the experiment itself, including the experimenter. We'll talk about this, towards the final weeks, that, there are ways for a salesperson or something to make you like a product more. And so we have to be very careful that that's not what this is about. And so people do neurophysiological research and other sorts of kind of sneaky experiments to make sure that these are in fact perceptual effects, and they absolutely are. People aren't able to detect things as well in terms of differences in custards and differences in these fruit drinks, if in fact the texture isn't a little different. And in this case, they're not even rating the how much they like it or how sweet it is whether or not these two custards are the same or different, and they might have a very difficult time doing so without the visible influence of the custard. And you can also do brain scans to make sure that when these effects are occurring, they're occurring in the part of the brain that you would expect a change in flavor to show up. So that's kind of another way of verifying it. And other things you find here is that overlapping cell groups there's a ton of cells in both the visual areas and the tasting areas that overlap with regard to taste and sight. So there's cells in the visual areas that are affected by taste, and cells in the taste areas that are affected by sight. And so certainly it looks like the brain is set up to very easily integrate what you're seeing into what you're tasting. And that can also be affected by smell too. There's tons of cells in the visual areas that are not only affected by what food you're seeing, but what food you're smelling. And as we'll see, these things can have these really interesting kind of cross-sensory effects to make your experience of food, very complex. But you might be getting, let's go back to the question of the dark restaurants, you might be getting a sense of what my experience was like there. Because now that you know that seeing your food has such a strong influence over how food tastes, you might not be surprised that it didn't taste great. So does food taste better in a dark restaurant? None of us thought it did. In fact, we all found the food very bland. All four of us. And we had, as I said, somewhere between 15 and 20 different dishes that we were tasting. Again, we all kind of had appetizers and main courses and desserts, and there was bread and there was beer that I had had before. Everything tasted bland to us. And if you look on Yelp for opaque restaurant, Santa Monica, two things. First of all, it's closed, sadly, I suppose. You can't have that experience anymore in Los Angeles. But secondly, if you look at the reviews on Yelp, the term bland, flavorless comes up again and again. And boy, if I were the chef for this restaurant, I would be heartbroken, because the chef is probably a very, well-trained, skilled chef that knows how to flavor food. But when you can't see your food, it doesn't taste as good. Now, we're going to be talking about an experiment having to do with blindfolding. Next class, I think, we'll be talking about touch and blindfolding experiments. And one of the kind of anecdotes that comes out of that research is that, we'll be talking about one experiment where people are blindfolded for 5 days, and they're in like a hotel room or something like that, and they're participating in touch experiments for 5 days. And they're getting their brain scanned and stuff. But one of the anecdotes that comes from that stuff is that when people are blindfolded and they have their next meal, one of the things they say is that food tastes especially bland. Okay? And it actually would taste bland for the first day or so, maybe even a little bit more. So if you aren't looking at your food, if you don't have the ability to see your food as in a dark restaurant, food does generally taste more bland. Okay? A lot of folks are interested. So does this mean if the blind have less vivid experiences of food? And the answer is no. What happens is, after a little while, and I think this even happens over the course of 5 days if you're blindfolded in that experiment, is that your other senses start kind of playing more of a role in your experience of food. So, I think, I don't remember how long it takes, but it's after like 3 or 4 days, food starts tasting good again. And you're probably using, your sense of smell, your sense of touch, and your sense of sound, sense of hearing that we'll talk about in just a second. But, I'm not surprised that there are so many reviews that use the term bland and flavorless, and there's a lot of reasons restaurants close. It looked like it closed about 5 months ago. Anyway, any questions about this restaurant, since sadly you won't be able to experience? They might have one in San Francisco. Maybe somebody will think about opening another one. It's really interesting. I don't name the restaurant in the book because I didn't want to, I didn't want to hurt their business. But I did say that, the food was very bland. People would ask me, should I go? And I would say, well, it's a really interesting experience. You go, and again, it's the darkest place you've ever been, and just to sit in a context that you're very familiar with a restaurant and not being able to see anything is kind of really interesting and cool. And you can, do little experiments with sound, and, see if you can, reach out and touch your friend's hands and all this kind of fun stuff. If you're going for the culinary experience, it's expensive, and it's like a high-end restaurant price, and you're not going to taste your food more vividly at all. Okay. Any questions about this? Okay. Let's go on then and talk about how another sense influences your sense of flavor, and that has to do with sound. And this was great because these experiments actually were done right before I started writing the book, and I was very happy to find them. And a little surprising, not too surprising that this is the case. I guess I'm surprised as to the degree that sound affects your sense of flavor. So one thing we know is that we judge crispness partly by what we're hearing. So the crispness of a carrot, the crispness of a stalk of celery, potentially a pretzel or something like that. We will say is based partly on the sound. Certainly it's how much do we have to, crunch it with our mouth and teeth, but what we hear will also influence that. We know that because of whether or not it's you chewing the carrot or your friend who's next to you chewing the carrot, your judgments of crispness of that carrot will pretty much be the same. So it's judged the same whether you're the one that's chewing or whether you're not the one that's chewing and you're listening to somebody chewing. It seems that in that context, your judgments of crispness are very influenced by sound. Now, what happens when we're eating, and how does that affect sound? Okay, so when we're eating, we are typically going through phases where our mouths are closed while we're chewing, and our mouths are open when we're putting food into our mouths. So in theory, you're getting the sound two ways. Okay? And this is something that the folks who design foods and how crisp they're supposed to be and all that sort of stuff is very aware of. Your sound actually at all times when you're making a sound or a sound's coming from your mouth, whether you're speaking, when you're speaking, we hear sounds coming from here two different ways. One is the normal way, where if I talk, sound leaves my mouth and goes around and yes, bounces all around Italy, all around my room, and comes back to my ear. So the sound leaves my mouth, goes around the air, it vibrates the air, and those vibrations make it back to my ear. That is one way. But we also hear sounds coming from our mouth another way, right? And that is called bone conduction. And what happens there is, for example, when your mouth is closed, you can just do this right now. Or you can just keep your mouth closed and just bang your teeth together gently. Like that. That sound you're hearing is not going through the air. That sound you're hearing is coming from the bone, is vibrating through the bones that exist between your mouth and your inner ear. And that's what this is showing here. And actually, it does more than that. It vibrates across your entire skull and then goes into the middle ear. And that's called bone conduction. And when we're speaking, we are always hearing our sound through bone conduction and most often air conduction as well. But you may have noticed that your voice sounds different on a recording than it does to yourself. I think it's pretty traumatic. When I was a kid, when I noticed that, I heard my voice on a little cassette recorder. We do like silly skits or something like that and record our voices. And the first time I heard my voice on a cassette recorder, I was like, oh my God, I sound so wimpy. And yeah, that's how your voice really sounds, right? When you hear your own voice, you hear it in a way that other people don't hear it. And it's through the bones that conduct the sound from your body through the mouth, the vocal tract here in your mouth. They resonate in your skull and then go to your inner ear. Other people don't hear that. Sadly, other people hear the whiny voice. Other people hear the thin voice that you hear on recordings. You hear your voice on a message on the phone or something like that. That's through air conduction. That's how everyone else hears your voice. Your voice sounds typically more resonant to you. Sounds a little deeper, maybe a little bit more authoritative even. And that's because you're hearing your voice through bones and not just through air. When you're chewing, you kind of hear both. You kind of hear through the air and through bones, okay? So it kind of changes as you chew. If you're chewing and you're talking, or you're chewing and you're putting things into your mouth, you are certainly getting that sound through the air. But if you're chewing with your mouth closed, as you're being polite, you're getting more of the sound through your bone conduction. So it kind of goes back and forth. But you hear enough through the air that this interesting experiment can be conducted. And this is an experiment, again, having to do with freshness or crispness of something you chew. The experiment is clever. What they did is they put people they put people in a sound booth so they could make sure that they could control all the sound. And they asked people, they say, you're going to sit in the sound booth, you're going to wear some headphones, we got a microphone in there too so we can get your responses. But what we're interested in is how fresh you think these different potato chips are. So we're going to give you different potato chips, a whole bunch of different brands, and we're going to ask you to rate its freshness and its crispness. Okay? Now, the truth is, all the potato chips that the subjects got were the same. They were exactly the same. How do you get the same potato chip to everybody? Pringles, right? They're the perfect sort of potato chip to do experiments with because they're all the same shape and size and all that stuff. Actually, I think what they did is they cut them a little bit, so every time they put it in their mouth, it was of a different shape, and they gave them the sense that these were actually different potato chips they were putting in. Okay, and they put people in the sound booth. There it is. Okay. They have a guy in a sound booth. They close the door, and they have a microphone in front of the person. They ask the person to chew. The microphone then picks up the sound of their chewing, goes to a computer, and then immediately puts a modified sound into the headphones that the subject is wearing. So the subject hears himself chewing based on a sound that is modified on the computer. The modifications are actually very simple. I don't even think you really needed a computer to do this, but they wanted complete control over the sound of the chewing. What they did is they essentially made the sound more trebly or bassy. If you know what those terms are, we talked a little bit about this, kind of a brighter sound or a duller sound. So the treble and bass controls you have on your car stereo. If you put the treble sound up, it sounds a little brighter. If you put the bass sound up, it sounds a little muddier. You turn down the treble and put the bass up, it sounds muddier. Remember, we talked about this when we were discussing how sounds travel through space. We were discussing echolocation and all that sort of stuff, right? The car gets close to you, and now you can hear the bright aspects of the music, not just the booming bass and the bass drum and all that when it's far away. So it was like that type of manipulation. So sometimes when people were chewing their potato chips, they heard a bright sound of themselves chewing. And then other times when they were chewing the potato chip, they heard a more dull, a little bit of a kind of a more bassy, kind of less bright sound. And depending on the sound they heard as they were chewing, they had very different ratings of the potato chip. If it was a louder, brighter sound, okay, it sounded crisper to them. It sounded fresher to them. They said, yes, I like the taste of this potato chip more. If, on the other hand, it was a dull sound, so they turned down the treble and turned up the bass, and it was a dull sound, they said, this is not a very tasty potato chip. I don't like this very much. This is kind of stale. I don't think I would buy this one again. And so, again, I know, it's always the same potato chip. It's a Pringle, cut to a different shape, but still, Pringles are Pringles everywhere in the world, right? But their experience of the crispiness, the freshness, and how much they like the potato chip was influenced by sound in this context. Cool. So now you know, okay, our sense of flavor, our sense of taste is influenced by sight, by sound. You learned last week it's very strongly influenced by smell as well. So that's also, of course, in the book, but it's an important thing to remember. Let's now talk about one last sense that it's influenced by, and that is touch. So I'm going to talk about a couple of kind of very dramatic examples of how touch can influence your sense of taste. But do know that we're going to then talk about much more subtle and much more common things, like the texture of custards and things like that, and how they can have a strong influence over how much, say, sugar we can detect even in things. But I'm going to talk about something a little bit more radical first, called tactile capture. And imagine this: you have a hard candy in your mouth, some sort of butterscotch candy or raspberry candy or something like that. Your favorite hard candy. It's got to be a hard candy. It can't be something that's going to dissolve quickly. It can't be something you're going to chew on, at least not right away. Okay, so there's a hard candy. And here's the thing about a hard candy is as it bounces around your mouth and bounces around your tongue, its flavor, first of all, is consistent, right? If it's a butterscotch candy, it's going to taste butterscotch all around your mouth. And secondly, its flavor seems to follow wherever the candy touches. So if it touches on the back of your tongue, that's where you say you can taste the sweetness. If it touches the side or like the blade right here, you'll say, yeah, the flavor is coming from my butterscotch candy and it still tastes butterscotchy and sweet. But you've learned this in other classes. That's not how our taste buds work. Our taste buds are not evenly distributed across the tongue, interestingly. So you might have learned this in high school or somewhere else, maybe intro psychology, I don't know, biology. We have our sweet taste buds primarily across the front of the tongue. Salt, the taste buds that are sensitive to salt back here. Sour, kind of more to the back side of our tongue, and bitter more towards the back middle. Okay? There are some folks that believe that there's more than just these four core tastes, one called umami. I don't know where those taste buds are thought to be, but they are not thought to be overlapping these at all. So when that hard candy is back here, okay, there are no sweet taste buds to taste the sweetness of that candy. It should taste bitter if there's a bitterness to it, and there probably won't be. It's a sweet butterscotch candy, right? But it still tastes sweet, even though we have no sensors for sweetness back there. Okay? And that's what's meant by tactile capture here. Food seems to carry its flavors with it as it bounces around our tongue, even though our tongue can't necessarily detect the flavors that we're experiencing, right? Which is really interesting. Somehow, where food touches our tongue, our experience is that we're getting the flavor from that location, even for candy, if we aren't having any of our sweet taste buds stimulated by the candy, which is interesting. Even a kind of extreme example of this tactile capture, I think you'll find interesting, called the sweet tongue illusion. It's a very simple idea. Here's what we do. We're going to do a more systematic experiment than just giving you a butterscotch candy and ask you, whether it changes its flavor depending on tongue location. What we're going to do is we're going to take a cotton swab, a new cotton swab. You unwrap it yourself. You dip it in sugar water. Okay, dip it in sugar water, and then pick it up, and we're going to ask you to do a map of your sweet taste buds on your tongue. Okay. Now, actually, I should mention your sweet taste buds are pretty much all here. They aren't on the sides. I might have said they were on the sides before. They're really, they kind of start right about here. Okay. So it's towards the very front of your tongue, very, very tip. That's where sweet is, right there. But what we're going to have you do first is take your little swab full of sugar water and put it on the side of your tongue, right? Say right about here. Hopefully you're seeing that. Yeah, okay, I'll make sure. There you go. Right about here. We're going to put it here. I'm going to say, okay, tell us if you can taste sweet. Now, it hasn't touched your sweet taste buds yet. So you put it here and you say, nope, I can feel it. I can obviously feel that it's touching my tongue, but no sweetness yet. I have not activated my sweet taste buds yet. And I start bringing this Q-tip around here. Nope, not yet. Not yet. I do not taste sweet yet. Not yet. Ah, there it is. I taste the sugar water now. Yeah, I bet we have some sweet taste buds right in the front. Yep, okay, let's write that down. Sweet taste buds in front. And now you continue to the other side of the tongue. Okay, so keep going to the other side of the tongue. Do you taste sweet still? Yeah, I'm still tasting sweet, no question. It is sweet all the way around here. Yeah, it seems weird, but I have no sweet taste buds on the, what is it? I guess left side from our perspective, left side of this tongue. All the sweet taste buds seem to be on the tip and on the right side of the tongue blade right here. All right, cool. That's kind of strange that you'd only have them on one side of your tongue. Let's confirm this. Take a sip of water, rinse out your mouth a tiny bit so we can start from scratch. Take the other side of your Q-tip, and now what we're going to do, dip it in sugar water, put it on this side of the tongue. Start with this side of the tongue. Okay. Do you taste anything? No, I feel the Q-tip, but I don't taste the sweet anymore. Anything? Nope. 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 Comes to the front. Oh yeah, there's the sweet again. Okay, let's keep on mapping it. Yeah, sweet, sweet, sweet. Very strangely, it seems that now this side of the tongue is sensitive to sweet, even though it wasn't before. Clearly what's going on, and it's not like the, I mean, one conclusion would be, for some reason, the taste buds hopped over my tongue in between these two trials. That's not what happens. You get the idea. Once those sweet taste buds are activated, where you are being touched by that sweet thing seems to track the sweetness with it. But it's not until these little guys are activated. Okay, so once it's activated in front, if this is the part of the tongue that's touched next by that Q-tip, this all seems like a sweet taste bud area. However, if you start here and that sweet bud area is not really the true sweet bud area, taste bud area is not being activated yet, it's not going to seem sweet until it is, and then wherever you're touching the tongue after that's going to seem sweet. So the sweetness follows where your tongue is touched, even though we're only sensitive there. So that's a dramatic example of how your experience of even the fundamental flavors of sweet, salt, sour, bitter, this all works with all these things, is influenced by where your tongue is touched after those taste buds are activated. Clearly, where your tongue is touched is very much going to influence how you taste things. Okay. What else? Texture. Texture strongly influences taste. So before I talked about the experiments where the texture that you're seeing, okay, the texture that you're seeing of a custard, for example, can influence whether it tastes sweet or not too sweet to you. If it seems like if it looks like a thick texture, I said before, it's going to taste the same exact, remember this is kind of sneaky little bowl, the same exact custard is going to taste less sweet than if it's a thin textured custard. But it's not just the view or the look of the texture that's going to make an influence. It is, in fact, how it feels in our mouth. So now we're actually doing something different where you can do this experiment with your eyes closed, in fact, without looking at the viscosity. And it turns out if you have a custard in your mouth that has a thick viscosity, it will have a weaker flavor, even though it has the exact same amount of sugar and chemical components than if that custard is made more thin. And that's clearly something that, any sort of company who, I gotta get an example of this actually. Any company who's making a food is going to have to take into account. This is something, all right, so I know this literature, I don't do research in this myself, until recently, because for my birthday, I bought myself an ice cream maker. I don't know if you guys have these things at home. They're great. Oh, they're so great because they're really inexpensive and they do an amazing job. I think I got the Cuisinart and it was like 30 or 40 bucks or something like that, right? And you have to kind of go by a little recipe and typically it's, milk, cream, and then sugar, and then whatever flavor you want, vanilla or something like that. Last night, in fact, I made a little frozen yogurt with banana and stuff like that. And when you start mixing it, you notice this, because what happens is, as it's being mixed in a bowl that had been frozen in the freezer for a long time. And so as it's being mixed over the course of 15 minutes, it starts off as a liquid, and then it starts firming up as the air and the frozen air is mixed into it. It starts getting thicker and thicker. And I should say, when you taste the liquid on its own before it starts mixing and getting hard, it tastes much sweeter. As it starts thickening up, it tastes less sweeter. And when it gets really cold, it tastes even less sweet. And we'll talk about how temperature affects taste in just a second. But this happened again and again. I've probably made 4 or 5 different things at this point with my kids. It's fun. They all come out really good. I mean, like artisanal kind of fancy pants ice creams. They're really good, and they're much easier and cheaper to do than to get in your car and go down. It's really good. I recommend it. But it's always been the same case. The mix, the fluid, the mixture that's liquid tastes so sweet. You have like a little spoonful or something. It's like, oh my gosh, this ice cream's going to be way too sweet. I'm going to throw it out. And then you let it mix, and it gets more firm. It gets thick. Ice cream can get very hard, and all of a sudden it tastes less sweet. And then the temperature is way down, and that's going to make it taste less sweet too. So I was so excited. I was like, not only have I made great ice cream, but I was able to demonstrate something I'd been teaching in my classes and I wrote about in the book. It was great. Big nerd. Anyway, temperature, as we've been discussing, also influences taste. And our tongues are most sensitive to flavors, including sweet and salt and bitter and sour and all the other flavors that we get through our noses, when it is at the temperature of our tongue. That is the best time to have a dinner, for example, right? So you want the food to come out, so at a restaurant, so the temperature that you're eating most of the food at will be about the temperature of your tongue, which is very close to 98.6. You take your temperature under your tongue, and that's 98.6. So when the temperature of the food is close to that, that's when things are going to be most vivid. If it's very cold or much warmer than that, things are not going to be vivid. And obviously, when you're eating ice cream, right, it's going to be much colder. But you don't even need a temperature drop that dramatic. What is that? It's like a 60-degree temperature drop from 98.6 to whatever it would be, 30 or whatever. Wait, not 30. I'm sorry, it's much lower. It's 0, right? My freezer is 0 degrees, I think. So it's a huge change. And so, things taste less sweet, that sort of thing. But even with a smaller change, very small change, 2 degrees higher or lower Fahrenheit can influence how strong a flavor is. So temperature has a humongous influence on flavor. And certainly, we send meals back at a restaurant if it comes out cold, right? It's unpleasant. I really don't like eating cold things that should be hot. I have trouble with leftovers. They have to be hot before I smell them even. But if I get a dish that's been obviously left out a little too long in the kitchen, it comes out a little cold, I'll send it back. I think a lot of people will, and you just feel like you don't want the unpleasantness of something cold in your mouth. But it's not just that. You can't taste the food as well, even with a drop of. So if it comes out to be, say, 95 degrees rather than 98 degrees, it's not going to taste quite as good, which is interesting. You can also do things with temperature to actually induce what's called phantom flavors. So it's one of these sneaky experiments where you give people 2 glasses of water, the exact same water, and one is very cold and one is very warm, or not, painfully warm, but warm. And you have them take sips of each. You say, I put a little salt in one and a little sugar in the other. Can you tell? And you say, it's very, very subtle. It's very subtle. I just put a little bit of each in there. Can you tell which one has the sugar and which one has the salt? The truth is, you didn't do anything to either of them. These are the same water, it's just cold and warm. people will almost always say that the cold water tastes salty and the warm water tastes sweeter, even though there is no difference. And it's like it's another kind of illusion. Your tongue cares so much about temperature that it will actually, to some degree, add its own flavor, it looks like in these cases. And it's not surprising. I mean, if you look at the brain, the parts of the brain that are, kind of sensitive to taste are filled with neurons that detect touch, right? So touch is a very, very, very strong sense to influence your experience of flavor, your experience of taste as well. So what do we learn? So it's sight, sound, smell, which is probably, I don't know, 75 to 80% of your experience of flavor is from what you're getting through your nose, and touch are all influencing your experience of taste and flavor. And so that's kind of the big story here. Out of all the senses we're going to be talking about, individually, so far we've gone through sound and smell, and now we're going through taste. Taste seems to be the one that incorporates or depends on, let's say it this way, taste is the one that depends most strongly on the other senses. It's what we say is the most multi-sensory of your senses. If you lose your sense of smell, like some of us did during COVID, food doesn't taste good. But now you also know, if we can't see our food doesn't taste good, right? Even the sound of our food is important, as is the touch. So without all those influences, we don't have much of an experience of flavor. And fortunately, we do have enough of an experience of flavor for us to love eating, going to nice restaurants, even watching cooking shows. That's what makes it pleasurable. So out of all of our senses, it's really the sense that depends on the other senses for it to be working well. All the senses are directly influenced on taste. You've already seen that. We've already shown how all the other senses influence our experience of taste. But they also kind of indirectly influence each other. And what I mean by that is, so for example, so indirectly means, the smell of something, the smell of, say, a custard, can influence how viscous that custard seems when we put it in our mouth. If it has a very strong smell, typically we will experience a custard as thicker when we put it in our mouths. And that thickness, in turn, that experience of thickness, in turn, will affect how flavorful it seems, how sugary it seems, right? And it's the smell that is affecting our experience of how thick it is, not the actual custard's thickness, but our smells affecting how thick it seems to us. And that experience of thickness is then influencing how sweet it tastes to us. So that's kind of an indirect influence of smell on taste. Because smell can affect our experience of touch, which is going to detect viscosity, which is going to affect our experience of taste. Other examples, the experience texture can affect how warm something seems to us, how cold something seems to us, and that can influence our experience of taste. So maybe this is something I experienced last night. So I tried. So I'm a big ice cream fan. I eat a lot of ice cream, and at my age, I got to be a little careful because we got to be worried about blood sugar and stuff like this. So I realize I don't eat a lot of ice cream when I make it. I just have a little bit every night. But I tried to find some recipes that didn't take so much sugar. So you're putting in like 1.5 cups of sugar into these kind of basic ice cream recipes. And this is not good. My doctor's not going to be really happy with this. So I found a recipe last night that's just no sugar. It's just a little honey. It's like a tablespoon or 2 of honey, and bananas, but the bananas got to be really ripe. So it's 4 really ripe bananas. That bananas get really sweet when they're ripe. And so I made this last night. It's actually really good. It's a frozen yogurt with Greek yogurt and banana, mush banana. Now, here's the thing about mush banana is it's really hard to get it consistent. So it's, kind of a pure, it's not consistent. There are some kind of banana lumps in there. I made it, they were very ripe, so I was able to squish them a lot. I had a little squisher thing. And then you put it in the machine and it does that too. But still, when you eat the ice cream, you can kind of taste that there are kind of, I don't want to say globs, it makes it sound unpleasant. It's not, it's really tasty. But there are, delightful pieces of the banana in the ice cream, in the frozen yogurt. That texture probably has, when I taste that, there's parts of the yogurt that have that thick texture, parts of it that don't have the pieces of the banana. And so when I taste the thick pieces, it probably influences my experience of the temperature. It probably seems a little less cold. And that can maybe make my experience of the frozen yogurt a little bit more vivid, a little bit sweeter. So again, here's an indirect influence of the texture of the banana that's in the yogurt affecting my experience of the temperature of the yogurt, all unconsciously, of course, which is going to influence, it's going to seem maybe a little bit warmer, which is going to make the flavor maybe a little bit more vivid. And these are all known, experimentally established examples of how your taste system, is influenced by so much, which is, why, I guess, being a chef, just even playing around with being a chef is such an exciting thing for so many people. It's like, having like these endless combinations. And I know typically we think about this as endless combinations of spices and things like that, but now think of all the different endless combinations we have, because all the other senses are so involved in this process of experiencing and loving food. So I think it's really interesting to think of it that way. Let's move on to, we've got 5 minutes still. Let's move on to another topic, another aspect of taste that has an influence. And I'm going to talk about this by introducing you to what's called a master sommelier. His name is Stephen Poe. And let me tell you what a master sommelier is. I don't know if you've ever heard of the term sommelier. It's French. That's my attempt at a French accent, sommelier. Right? And they are the people at high-end restaurants that are there to recommend wine. And they're also the ones that purchase the wine for the restaurant. They decide to, these would be a good, collection of Merlots. These would be a good collection of Zinfandels to have at this restaurant. They know a lot. And they can recommend, okay, if you're having fish, it's a very strong tasting fish, so maybe you should have a Zinfandel. It's a very robust flavor for a wine. These folks know a lot. That's a sommelier. Okay? Probably Opaque had a sommelier. We didn't have wine that night, but if we did, they might have come over and told us a little bit about the wine and which ones they recommended. I'm not much of a wine drinker. I'm more of a beer drinker. The folks that teach sommeliers how to, do this correctly, they teach them about wine. They teach them about, the different types of wines that are local to their, region, that sort of thing, are called master sommeliers. They're like the big teachers. And I haven't looked this up recently, but about 3 years ago, there were only 216 master sommeliers in the world. And they all were trained at this one place in France. And then they do a lot of training on their own. But they are trained and then tested to be certified as master sommeliers at this place in France. Okay. How do they become master sommeliers? It is not easy. They take 3 exams to become master sommeliers. And at least one of those exams takes many, many years to pass. The first exam is kind of straightforward, kind of like studying, out of a book sort of thing. They learn about wine theory. They learn about the different production techniques, that sort of thing. How to grow different grapes, that sort of information. And that's, paper and pen tests, and most of them pass that. If not the first try, they get it the second try. The second one's serving decorum. So that's the appropriate way to serve wine at a restaurant. I don't know if you've ever been in a situation where they bring over the wine. Sometimes it's the waiter or something, but if it's a high-end restaurant, it's the sommelier person. They bring over the wine, they pour it in the glass, they have you sniff the cork, all these sort of things. They tell you about the wine, all that. And there's actually a lot more to it. And they also, have to know other things about serving decorum. And that they have to study for that one as well. But the one they study for the most, and the one they have to take over sometimes 3, 4, 5 different times, and they can't take it more than once a year, is what's called the blind tasting. And it works like this. They are given 6 wines that are put in front of them in glasses. And they're either going to be all red or all white, typically all red. Okay? And they can be from anywhere in the world. And it's the job of the master sommelier student, the one that's trying to pass the exam, to sip them and tell, I think, 3 judges what the grape variety is, what region of the world the grapes have been grown in, and what the vintage is, meaning when it was bottled. They have 25 minutes to do this. They have 25 minutes to be correct with their answer, as well as conveying their reasoning and a vivid description of the wine so they reach that conclusion. And they're judged by 3 masters. We will talk about how Stephen Poe was able to eventually pass this test next class. Okay, see you Thursday.